How do we know when we are live?
We are live. Okay, hello everyone. Good afternoon, morning, evening, depending where you are, and welcome to the new episode of Neural Development on the Worldwide Neuro Network. I'm replacing Denis again as a host, and I hope wherever he is, he's having a fun time, but also connected and listening to us. Denis, if you're there. Okay, so welcome everyone. It's a really great pleasure to introduce Steph Baulac today as our speaker. Steph is a group leader here at the Institut du Cerveau, the Paris Brain Institute. And she's, in addition to being a fantastic scientist, a team leader, a great mentor to her students, she's also really involved in the life of the Institute. She's a member of the Scientific and Medical Strategy Committee at the Institute. She's also the scientific head of our sequencing facility. She's very involved with French associations in research on epilepsy, and so she's really an active person in every way. She's at a personal level, I mean, I consider her a friend. She has a fantastic sense of humor, which is really cool. And so Steph did her PhD right here at the Pitié-Salpêtrière Hospital in the team at that time led by Yves Agide, with the supervision of Alexis Brice and Eric Le Guen. And then she did a postdoc at Harvard Medical School with Dennis Selko, so really sort of illustrious PhD and postdoc time. She did great in both. Came back to France as a senior researcher and became, I think in 2013, team leader and research director at the INSERM, and then team leader at what was now to become the Paris Brain Institute, or the Institut du Cerveau, where she is still today. Steph is probably one of the 5 best unquestionable top labs in genetics of epilepsy, and what I admire about her work is how she really combines the clinical aspects, including clinicians associated with her team, going all the way from the patient, getting to understand the sample, finding the genetic cause, modeling the disease in the mouse, and going into the cell biology and molecular biology of what's going on. So she's really spanning the entire spectrum, trying to understand these disorders with particular focus on focal epilepsies. And I just want to highlight a couple of her papers that are my personal favorites. In 2013, I think, she had a paper in Nature Genetics where she discovered that a gene involved in the mTOR pathway called DEPDCS was involved in focal epilepsies, and because those mutations were in single copies, it was thought that it was an autosomal dominant disorder, which then Steph went on to show in a really beautiful piece of work that I'll put the link in a second, that in fact somatic mosaicism in the brain of these patients combined with a congenital mutation explained many of those focal epilepsies, and linked all of that to the phenotypes of these neurons, which have these sort of big balloons. I guess you will see them in the talk today. I don't know, Steph, are you going to show some of those? Sure. Balloon-like cells that turn out to have this sort of double mutation. And so now the big question is, what is autosomal dominant? What is second hit? What's going on? I hope we'll hear from Steph about where we are in the field today. So it's a pleasure to introduce you. It's an even greater pleasure to see you and work with you on a daily basis. And the floor is yours, Stephanie, and whenever you're ready.
Okay. Hello, everyone. I'm very pleased and honored to be part of this webinar series. Thanks a lot, Bassem, for the nice intro, and thanks, Denis, for the invitation and the teaser on Twitter. Ah, no. Hey, we did repeat, huh?
Voila.
Voila. Are we okay?
It's all yours, so I'm going to close my video and audio, and we're ready to go.
All right. So I've seen on this Twitter teaser that the main question was, what is the hippest cocktail currently? So I would like to share with you one of these cocktails that I've seen in Paris currently, the Blue Raoult, and I will not comment more on it. Okay, so let's get started. So our lab has a long-standing interest in epilepsies, and we have been shifting our interest more recently to the field of malformations of the cortical development. So today my talk will tell you a bit the story about this continuum between non-lesional epilepsies and lesional epilepsies with the navigation from inherited germline variants to somatic de novo mutations. So we'll start with more or less traditional genetics, but I promise to move on to more neurodevelopmental aspects and disease modeling, which I guess this crowd is more interested into. So our main goal is to understand the causes of focal epilepsies. And the etiologies are quite diverse. They can be of genetic origin, they can be structural due to a brain tumor or brain trauma, they can also be autoimmune or metabolic. And the second aspect that we are looking at is to understand the impact of disease-causing mutations using 11 disease models. So we are, our research is based on familial forms of epilepsy, which is a way to identify new actors and new pathways in an unbiased way using whole exome sequencing strategies or whole genome strategies. So we have many collaborations with clinicians for patient recruitment. These are on-site clinicians, but also at the European and international level through different consortia of genetics of epilepsy. But we also take advantage of using epileptic brain resected tissue. And with this, we have a very nice collaboration with the neurosurgery department of the Fondation Rothschild for child recruitment. But we also have some adult recruitment at the Pitié-Salpêtrière and Sainte-Anne Hospital in Paris. To better understand the pathogenic mechanisms of epilepsy, we use mice model. And we have recently implemented iPSC brain organoids, but these data are still too preliminary to be presented here. So epilepsies, so they are frequent, they are heterogeneous, and they are characterized by spontaneous and recurrent seizures. And these seizures reflect a hypersynchronization of neuronal networks. And among genetic forms of epilepsy, there are four major phenotypes. The genetic generalized epilepsies, and these are usually polygenic. Then the non-acquired focal epilepsies, and that's the Mendelian forms of focal epilepsies that are isolated with a very clean MRI. These have an autosomal dominant transmission. Then we have the lesional focal epilepsies. They are sporadic, and these are the ones caused by somatic mutations. So this will be the big focus of my talk today. And then the group of the developmental and epileptic encephalopathies. So with this, we cover different mechanisms of genetic mutations and mode of inheritance. And so the genetic architecture of epilepsy is declined into different scenarios. Traditionally, multiplex families with inherited mutations and a phenotype transmitted over the generations have allowed the identification of mutations in different ion channel genes, neurotransmitter genes. And with this, the epilepsy has entered the world of channelopathies along with cardiomyopathies. And so for many years, epilepsy was exclusively considered as a channelopathy. But this challenge has been, this notion has been challenged 10 years ago with the advent of the high throughput NGS technologies that have allowed the identification of hundreds of genes, especially in developmental and epileptic encephalopathies. So these forms of epilepsy are very severe, they're sporadic, and the way those genes have been identified is through parent-child trio analysis using whole exome sequencing and the identification of de novo germline mutations. But now more recently, epilepsy has joined the world of brain somatic mosaicism, especially in the sporadic lesional focal epilepsies and the discovery of the somatic mutations arising during neurodevelopment and affecting only a subset of cells. So today I will show you how we have discovered a bit this continuum between non-lesional and lesional epilepsies. So let's step back in 2013 when our team and an Australian team discovered a new gene called DEPDCS5 for DEP-domain containing protein 5. And at this time, the function of the gene was totally unknown. We only knew that it had no homology with any ion channel or neurotransmitter gene. And with Saiko Ishida, a postdoc in the lab, we had performed whole exome sequencing in different affected individuals of this family. And this family had one characteristic. First of all, there were some asymptomatic individuals, as you can see here. And the phenotype was a bit variable because epilepsy could emerge from different brain area in different family members. And in addition, we had this individual, which was a bit enigmatic for us. At that time, he had a lesional epilepsy with a focal cortical dysplasia. And at that time, we were not sure whether this was coincidence or really due to the DEPDCS5 mutation. And with my colleague Fabienne Picard in Geneva, she had collected a cohort of 13 families with autosomal dominant focal epilepsy, and we were able to identify mutations in 6 out of these 13 families. Most mutations were loss of function, nonsense, and frame shift mutations. So we didn't know the function of the protein, and so one thing we could do is to assess whether those stop mutations would act as dominant negative and through a truncated protein, or whether they would behave as haploinsufficiency due to the degradation by the nonsense mediated decay system. So the NMD system degrades any transcript harboring premature stop codons. So to assess this question, we cultured lymphocytes from different patients with different mutations in DEPDCS5, and we cultured them with or without M18, an NMD inhibitor. And what we can see here is that the mutation that is present in the genomic DNA was not detectable anymore in the transcript in absence of M18, indicating that this transcript is indeed degraded. And this was showing a haploinsufficiency mechanisms, and we could show that in many other patients. However, in one patient that had this mutation located in the last exon of DEPDCS5, and as expected, we could see that this transcript escaped the degradation by the NMD, and there's probably a shortly truncated protein in this patient. So with this, we learned that the relevant disease model for this DEPDCS5 related epilepsy is a model of haploinsufficiency, of loss of function. So shortly after our discovery that DEPDCS5 mutations were involved in focal epilepsy, the group of David Sabatini at the MIT could elucidate the function in vitro of DEPDCS5. So DEPDCS5 is part of a complex called GATOR1, together with NPRL2 and 3. This complex is also inhibited by another complex called GATOR2, and GATOR1 is a repressor of mTOR-C1, so the complex 1 of the mechanistic target of rapamycin. The protein DEPDCS5 itself has a GTPase activating protein activity, exactly the same than the TSC2, one of the 2 causative genes in tuberous sclerosis. And tuberous sclerosis is a multi-organ disorder in which patients very often have severe epileptic seizures. So this complex mTOR-C1 is a serine-threonine kinase that phosphorylates many proteins in the organisms. And among these 2 proteins, 2 are well known, S6 and 4-EBP1. And these 2 substrates are usually are commonly used to monitor the activity of the mTOR pathway. And we will use them later during the talk. So mTOR is a hub in the cell. It regulates key functions in the cell, such as cell growth and cell proliferation, by acting on protein synthesis, RNA translation, and inhibiting autophagy. This is a more complex view of the pathway, which is more close to the reality. mTOR is actually a sensor of different nutrients, can be glucose, amino acids, growth factors, neurotransmitters. And the classical way of activation of the pathway is through the PIK3, AKT3, TSC1 branch. But in our case, in the GATOR1 branch, it is specifically regulated by amino acids, and in particular, leucine and arginine. So we are far away from ion channels and our classical epilepsy genes, and we were quite surprised to find mutations in such a ubiquitous pathway, causing an isolated focal neurological disorder. So next to this, with Sarah Wekwisen, a colleague of mine, she was working in Paris in the lab at that time, we asked whether mutations in the other GATOR1 and GATOR2 encoding genes might explain some of the DEPDCS negative patients. So for this, we designed a targeted gene sequencing panel and sequenced 90 patients with various focal epilepsies. And we were able to find other mutations, again, stop mutations, in both NPRL2 and 3. And this was also described by other groups. And so overall mutations in either DEPDCS5, NPRL2, or 3 will lead to a similar phenotype with focal seizures. So we cannot talk rather about the GATOR1 phenotype spectrum. So during the past years, many reports have published new mutations in DEPDCS5 and in GATOR1 encoding genes. And so with Sarah Baldassari at the lab, we aim to further refine the phenotypic spectrum associated to these mutations. So we collected a large cohort of 73 families, more than 100 patients, and we could show that in most cases, they have focal seizures, mostly sleep-related hypermotor seizures, with an early adjunct onset. And importantly, many patients are drug-resistant to antiepileptics, which is a major clinical issue. They also have frequent comorbidities with cognitive and psychiatric disorders. And we also realized at that time that patients with mutations in DEPDCS5 or GATOR1 in general had a higher risk to SUDEP. And SUDEP is the sudden and unexpected death in epilepsy. It can occur in children, but also in adults. But we also found that 15% of the patients of the family had a focal cortical dysplasia that was diagnosed by MRI. So here we started to understand that that was not coincidental to find those malformations of the cortical development among those patients. So what we were, the question that we were asking at that time is, how can we explain such a variability in the phenotypes? With inactivating DEPDCS5 mutations, we can find asymptomatic carriers, or patients with more or less mild non-lesional focal epilepsy with this variable foci. And at the end of the spectrum, much more severe epilepsy, refractory to antiepileptics, and with this lesion that can be seen at the MRI. And more intriguingly even, within the same family, those different phenotypes can be encountered, while the same DEPDCS5 mutation is inherited. So now that's when we enter the malformations of the cortical development. As you know, the cortical development is a highly regulated process, and migration defects can lead to these malformations of the cortical development. They have a genetic origin. Some of them can be lissencephaly, heterotopia, microcephaly, polymicrogyria. And in most of these cases, it's de novo germline variants in several genes, mostly encoding tubulin and associated proteins that are involved. And focal cortical dysplasia also belong to the spectrum of malformations of cortical development, but they are very focal. They can be very small, barely detectable by MRI, affecting only the bottom of the sulcus of a gyrus, for instance, but they can be also more extended and sometimes can even occur in a whole hemisphere. And this is what we call a hemimegalencephaly, or HME. FCD is a major cause of pediatric focal epilepsy with the developmental delays associated. And the important point is that the majority of the patients are drug resistant. So the only therapeutical option to treat them is to perform a resection or a functional disconnection of the epileptogenic zone in the affected hemisphere. And currently, FCD represents a large proportion of the whole epilepsy surgery population. And so the etiology of FCD had been unknown until recently. But for some reasons, genetics causes were thought to be unlikely. So now let's have a look at the neuropathological features of FCD. The International League Against Epilepsy has classified them into three main groups. And here we have FCD type 1 and FCD type 2. And in both types, there's a cortical dyslamination, and there's a blurred white matter cortical junction. But what is interesting in FCD2, and I find it very fascinating, is the presence of normal cells, such as dysmorphic neurons, shown here, and balloon cells. And so now my talk will focus mostly on FCD type 2, which are the ones associated with DEPDCS5 mutations. So what do we know about those dysmorphic neurons and balloon cells? Dysmorphic neurons are cytomegalic cells. They are huge. They're big. They can be pyramidal mostly, but some interneurons can also be cytomegalic. When looking at them in the tissue, they are malorientated, and they're characterized by a high content in neurofilament. So one way to stain them is to use the SMI32 marker. One hypothesis about their immaturity is that there might be some remnants of early subplate cells. And the group of Carlos Cepeda has done a lot of work on physiological work on human FCD tissue, and he has shown that those cells are probably hyperexcitable and that those dysmorphic neurons might be pro-epileptogenic. In contrast, the balloon cells that are shown here, they are huge. They are really bizarre cells with a glassy cytoplasm. The nucleus is often eccentric or sometimes binucleate, with two nuclei. And they have a mixed lineage origin. They express both glial and neuronal markers, and of course they are immature. And one hypothesis is that they may represent radial glial cells. They have no axons, no dendrites, no synaptic inputs, and they have been shown to be electrically silent. So to date, the developmental origin and the molecular identity of both cell types remains quite enigmatic. Now, if we look at a tissue from a DEPDCS5 resected brain specimen, this is here those dysmorphic neurons, and they are stained with phosphate-6, one of the substrates of mTOR. So what you can see here is that those dysmorphic neurons have a hyperactivation of mTOR, and this is expected because DEPDCS5 is a repressor of the pathway, and so a loss of function of the repressor will lead to an increased activation of mTOR. But what is very interesting is that you see that these neurons are surrounded by normal neurons. So there's really a mosaic patterning of mTOR activation. And this is what led us to hypothesize that there's some brain mosaicism occurring in these patients. So what happens when somatic mutation arise? Well, either it occurs very early during the development, and it might affect different organs in the body. But it can also occur after neurulation, and then would affect the whole brain or part of the brain or only a small part of the brain, depending on the timing when the mutation occurs. And we now know that even at the individual level, neurons carry somatic mutations, and that those mutations even accumulate with aging. So this brain mosaicism was the work of some pioneer labs such as Chris Walsh, Joe Gleason, John Hawley, Bill Dobbins. And at that time, in 2012, these mutations were found in overgrowth syndrome and megalencephaly syndromes. And so we were very far away from our familial non-lesional focal epilepsies. And the link was not obvious, even now it seems obvious. So in the context of DEPDCS5, we have those inherited mutations transmitted over the generations. And sometimes one individual has this FCD. So what we hypothesize is that according to the nascent two-hit model that is used in oncology, we saw that maybe there was a first hit, the germline mutation, a point mutation, affecting one copy of the gene, and that a second hit occurring in somatic cells would inactivate the second copy of the gene, and that the cells carrying both mutations would be at the heart of the lesion and explain the focal cortical dysplasia. And depending on when the mutation occurs, the second hit mutation, this would drive the size of the lesion. So to test this hypothesis, well, we need brain tissue. that's the only way to check this hypothesis. And epilepsy and gliomas are one of these unique neurological disorders that get operated and give us a direct access to tissue. So we initially started with this family, and this male here had been operated more than 20 years ago to treat his dysplasia. And so we have been searching in the archives, and we finally found the block of FFP tissue from this patient. But of course, this tissue was very old, was formalin fixed, paraffin embedded, and the quality of the DNA was very poor, and DNA was pretty much fragmented. So we couldn't amplify it through NGS technologies. However, we were able to perform PCR and Sanger sequencing. And so this is what we found. This is here the DNA derived from the cortex, from the FFP sample, and derived from the blood. And this is here the germline mutation in DEPDC5 that is segregating in the family. And so we sequenced the whole DEPDC5 gene in the cortical sample. And finally, we found here a double peak here, indicating a mutation, a nonsense mutation that was not present in the blood sample. So that was really the first evidence that indeed this hypothesis of a second hit might be true. However, because of the limitations of the quality of the DNA, we were not able to assess the mosaic level of this somatic variant, and we were not able to prove that both mutations are in transconfiguration and therefore leading to a biallelic inactivation. So that's when we realized that we really needed some fresh tissue. And we initiated this collaboration with the neurosurgery department of the Fondation Rothschild, led by Georges Dorfmüller. And they operate approximately 30 children to treat their seizures per year. And so I've been conducted this work with Mathilde Chipot, and we have collected approximately now 200 surgical cases of children aged between the age of 3 months and 16 years of age. And we collect blood samples, the resection that we immediately snap freeze in the surgery room, and we also try to collect some CSF for further studies. I would like to show you a quick movie of a surgery, so sensitive person may close their eyes for a few seconds. So everyone in the team often has to go to the surgery room, and so we can watch the whole resection and cut it at the surgery room when it's big or when we want to define what is really the heart of the lesion and what is more peri-lesion. And that's it. Okay. So in this, with this, we also collaborate hand in hand with the neuropathology department, because as you will see in my talk, there's a very strong link between the neuropathology findings and the genetic findings. And before sequencing anything, we always have a look at the piece of tissue that we get, the frozen tissue, which might be different from the tissue that gets to the neuropathology, because of course it's an adjacent block. Ça recommence. Bon. So what Sarah Baldassari, postdoc in the lab, did is to design a study to search for brain somatic mutations in those FCD samples. So we selected 80 surgical cases with either a small FCD or a larger dysplasia in the context of an HME. And she designed a targeted gene panel with the idea to sequence some genes of the GATOR1 branch, but also a few other genes of the mTOR and PIX3CA pathway. And the idea was to have a small panel, but to be able to reach a high coverage, to be able to detect any low allele frequency somatic variant. And for this, we sequenced paired samples, brain and blood, to be able to do a subtraction and to identify brain-only variants. So detection of somatic mutation is not easy, and we have to use dedicated bioinformatic tools. And here we have been using MUTEC2, BARScan, or MPilot, an in-house developed tool. And the idea was to be able to detect any variant present at least at 1% of the mutant reads. And all the variants that we identified had then to be further validated by alternative sequencing strategies. And we have been using ultra-deep site-specific amplicant sequencing and digital droplet PCR. So these are the results that we have obtained. We were quite surprised, but we found a shower of somatic mutations in the FCD2 cohort. So 60% of the cases were solved after this first screening. And we found mostly single-hit mutations in PIK3CA, AKT3, REB, but mostly mTOR. Many patients were actually mutated in the gene mTOR itself. And those variants are all recurrent hotspot variants, and they have already been found in some other cancer. So those variants are well known, and they are known to lead to a hyperactivation of mTOR. We also found in this cohort 5 patients with a DEPDCS5 mutation, with a germline mutation, and we were looking for the famous second hit, since according to our hypothesis, in repressors of the pathway, the 2 hit is necessary to conduct to the lesion. So just the overall data of this cohort, we also found a nice correlation between the rate of mosaicism and the size of the lesion. And so in small FCD, as you can see here, the mosaic level was quite low, in average 5%, but it could be as low as 0.3%. to explain this severe disorder, which was quite surprising. And this is also showing that you really need deep sequencing strategies to identify these variants. In intermediate FCDs, variant allele frequency ranged from 5 to 10%. And in HME, the biggest ones, variant allele frequency ranged from 10 to almost 40%. So we also realized that it's not the same variant that we find in small FCDs and HME, and this is probably due to the nature of the variant itself, where we believe that some variants are more activating of the pathway than others, and probably that those mTOR variants are probably lethal if found at higher rate of mosaicism. So with this, we could also show that there's actually a real continuum between FCD and hemimegalencephaly, that it's the same disorder. It's just a question of mosaic rate, and that this depends on the developmental time and progenitor cell types in which the mutation occurs. So now, back to our DEPDCS5 story, we were looking for the 2-hit mutation. And so actually, we could, after this screening, we could elucidate and find the second hit in only 1 patient. This was a 6-year-old boy with a small FCD2A, and he had a germline mutation in position 286. And we were lucky enough that the second mutation, the somatic one that we were looking for, was very close to this one. And with this, because both mutations were very close to each other, we could amplify them in a single amplicon and perform ultra-deep sequencing. And when I say ultra-deep, that was really like 200,000x. And why did we do that? Well, we wanted to make sure, first of all, that the mutation is never present in the blood, that it's brain-specific. And this is the case. This is a somatic mutation here. And the red dots are indicating when the mutation is present, and we never find it in the blood. And when we look in the different reads from the sequencing of the DNA derived from the resection, we could show here that in the reads carrying the germline variant, the somatic mutation is never present. So this was finally proving that indeed both mutations are in transconfiguration, leading to a biallelic inactivation of the DC5. And what was very interesting here also is that this resection was quite large. And that day, the neurosurgeon gave us several pieces of tissue. One piece that he found very dysplastic and what was really at the heart of the epileptogenic seizure onset zone. And here we found 10% of mosaicism of the somatic variant. But in the perilesional piece of tissue, we found only 0.3% of mosaicism. So there is a nice gradient of mosaicism in the brain with higher levels in the epicenter than at the border. So the next question that we asked with Sarah Baldassari was, what is the cell origin of these variants? So to do that, we used again frozen tissue from FCD patients. And what she did is to microdissect pools of cells. And she microdissected some dysmorphic neurons, some balloon cells, some apparently normal neurons, at least based on their morphology and their size, their soma size. And we also sequenced some glial cells. Actually, we did not sequence them. We used digital droplet PCR, which is a very sensitive and accurate technology, allowing to assess the presence of a mutation in a droplet base, so in unique molecules. And this is what she found. So this, the first tissue she has been working on was an FCD type 2B, carrying an mTOR mutation. And that's the level that was in the bug tissue, so approximately 5% of mosaicis. And when she checked normal neurons and glial cells, she could almost not detect the variance. But when looking in the dysmorphic neurons in blue and the balloon cells in pink, we saw that the mosaic level was nearly 50%. This is indicating that nearly all dysmorphic neurons and balloon cells are mutated. And this, of course, is a nice explanation why they are cytomegalic, because they have this mTOR activating variant that is increasing their soma size. But then she also looked at an HME patient, so with a large dysplasia occurring the whole hemisphere with a PIK3CA mutation. And here the VAF in the bug tissue was higher, was 20%. And in this case, she could detect also the mutation at quite high level in the glial cells. And so here, this is indicating that here, as expected, the mutation has occurred much earlier in probably neuroglial progenitors, while in the FCD2B, much smaller, it was in neuronal progenitors that the mutation has occurred. So with this technology, combining microdissection, laser microdissection, and digital droplet PCR, we sought to use this approach to search for the second hits in the patients where we didn't find it. So we knew that the other patients had no single nucleotide variants, and we suspected that maybe structural variants may occur. So we had then access to this hemispheric dysplasia from a 3-month-old girl, and she was carrying this germline mutation here. So Sarah did again some pools of glial cells, normal neurons, and dysmorphic neurons. There were no balloon cells in this tissue. And this is the results of the sequencing from the DNA derived from the blood. And as you can see, we have this double peak here with the Y-type allele, the G, and the mutated allele, the E. We can detect this double peak in glial cells and in normal neurons. However, in the dysmorphic neurons, as you can see, the Y-type peak has almost disappeared. And when we looked at a more global view, we could indeed see that at least a huge part of the chromosome 22 was lost. So this is a mechanism of loss of heterozygosity. And we suspect that this mechanism is occurring much often than we suspect, exactly as in oncology, but we need specific tools, and this is not detectable by sequencing technologies. So now what about the unsolved FCD2 cases? So as I said, we have mutations in 60% of the cohort, and 40% we didn't find any mutation. So we first asked whether they might also be linked to mTOR activation, and so we performed immunostaining with phospho-6 marker in all the tissues. And what we can see here are the panel negative cases, and here few mTOR patients. And when we compare the staining, you can see that it's very similar. So for me, there's no doubt that those patients also have mosaic variants in mTOR pathway gene. And now the question is whether those causative genes were not included in our panel, or whether it's undetectable mutations like CNVs or structural variants, or maybe it could be due to an ultra-low somatic mosaicism. So this is something that we also assessed in the different tissues. And what we have been able, so we have quantified the balloon cell density in most of the tissues. And the first thing to know is that this density is highly variable. In some tissue, you may find only 2 balloon cells, which is sufficient to have a diagnosis of FCD type 2. But in some tissues, it's full of balloon cells, like 400 per section. And also this density may be variable between FFPE and frozen tissue, FFPE for diagnosis and frozen for research purposes. And what we can see in this graph is that actually the panel positive cases all had a higher density of balloon cells than the panel negative cases. So this indicates that probably we didn't find the mutation because we were underneath the threshold of detection of those variants. So one way to achieve this is probably to enrich with balloon cells and dysmorphic neurons and then perform panel sequencing on microdissected pools of cells. So very recently, one of the patients that was in our cohort, it was an 18-year-old woman. She died of a SUDEP, of a sudden unexpected death in epilepsy, and she was carrying a DEPDCS germline variant inherited from her asymptomatic mother. And so she had an MRI that was negative, EEG negative, and not really a focal seizure onset that was well delimited. So we had access to the postmortem tissue, and with OMA-BSET, we actually screened different brain areas, and we found actually few sparse dysmorphic neurons labeled here by phosphor6 or SMI32. And these were not detectable by MRI, probably because the lesion is too small. But now this is raising a bit new concepts in the field, because so far we saw that heterozygous mutations were explaining non-lesional cases, and that the second hit would explain the focal cortical dysplasia. But now we start to wonder whether the presence of germline mutation may facilitate the occurrence of a somatic mutation, and that maybe the somatic hit is also present in all DEPDCS cases. And maybe this would be enough to explain why these patients have variable foci, which was also an unexplained point in our families. So I would like also to underline that, of course, our team is not the only one contributing to these advances, and that many other labs are also working and having a very similar data. We are also very happy that the second hit DEPDCS hypothesis has been confirmed by other groups now. And also highlight that, so epilepsy, as many other neurodevelopmental disorders, is now joining this brain somatic mosaic network together with autism, schizophrenia, bipolar disorders, etc. And the reason why this genetic was hidden before is really a matter of access to the brain and deep sequencing technologies. So I am almost done now with the genetic part. But before moving to disease modeling, I would like to have a look with you on the tissue. So this is an FCD2 tissue from an adult patient that was recruited by Francine Chassou at Sainte-Anne Hospital. And this tissue was actually nice because there was a clear cluster with dysmorphic neurons, staying here with phosphor6 and UN, and this is a zoom on those dysmorphic neurons. And in another area of the tissue, there was a cluster of balloon cells. Usually those two cell populations are intermixed, but here they were clearly clustered in different areas. And this is a zoom on one of those balloon cells. So with Jean-Christophe Poncer at the Institut du Fer-et-Moulin in Paris, he performed multi-electrode array recordings on acute brain slices of this tissue. And MEA, performed in different brain areas, has revealed different activities. So there was actually one zone where there was interictal-like activity, and with a gradient of spontaneous multi-unit activity or high potassium-induced multi-unit activity. And here in this area, there was no activity at all. So what Théo Ribière did in the lab is to perform some phosphor6 staining and to match them with the activity. And what we found here is that where the highest activity is recorded, that's clearly where the density of dysmorphic neurons is the highest. He measured the density of those neurons in every part of the cortex. And there was this gradient with less dysmorphic neurons and no more here. But what is interesting is that in the cluster with balloon cells, that's where there were no activity. So this is confirming a bit for the first time all the work of the group of Carlos Cepeda, showing that those balloon cells are clearly silent, and that's not where seizures may emerge. So now let's move to preclinical mouse models. And this is the work of Theo Rivière, doing his PhD in the lab, and Alexandre Bach doing his postdoc. So we are back to DEPDCS5 and the second hit. At that time, we wanted to prove that a biallelic inactivation of DEPDCS5 in a subset of cells is sufficient to recapitulate the features of FCD at the neurodevelopmental aspects, but also clinical aspects with seizures. So what Theo did is to design some guide RNAs, two independent guide RNAs, and to combine them with CRISPR-Cas9 genomic editing in order to delete DEPDCS5. And we performed in utero electroporation at hit 14.5 in the ventricles, and this way targeting layer 3 pyramidal neurons. By fact sorting, he was able to show that only 1% of the cortical neurons were indeed CRISPRized by this way in the electroporated zone. So very few neurons are actually biallelic inactivated for DEPDCS5. So he first assessed whether those, we can see a migration defect, and he first checked at the age of E18 for the migration. This is the control condition where the GFP neurons have migrated to the cortical plate as expected. But in the focal knockout condition, in green are the CRISPRized neurons. You can see that many of the cells are retained in the ventricular zone or a subventricular zone. He then administered Rapamycin, an mTOR inhibitor, to the pregnant dams 1 day after the electroporation. And here you can see that this totally rescued, prevented the migration defect in the cortex here. Okay. When we look in the adult brain, there's still a more subtle defect of migration. You can see here the control where those GFP neurons are now in the layers 2 and 3, and they are more retained in layer 4 in the two knockout conditions with the two different guides. We then asked whether a deletion of DEPDCS5 would increase the activation of mTOR. And so for this, we performed some phospho-6 staining. This is performed in adult mouse brain. These are the two conditions, guide 1, guide 2. And what you can see here with the phospho-6 staining is that indeed there's a more intense staining of phospho-6, and more cells. But importantly, you can see by eye that those cells are much bigger than in the control situation. And this is, again, what is expected with an mTOR activating deletion to enlarge the size of the soma of those neurons. Oh yeah, I have a fancy movie to forget this. So because after the talk of Alain Chadotal, you know, I thought I really needed me to a cool movie. And this is to show you how those dysmorphic neurons are in the cortex surrounded by other normal neurons. It was a bit fast, but I'm sure you can appreciate that those neurons are much bigger than the surroundings. So then with Charlotte Deleuze at ICM, we aim to assess the electrophysiological and morphological properties of those CRISPRized neurons. And so she performed some recordings and biocytin filling of layer 3 pyramidal neurons. This is here the reconstruction of a control condition, and this is of a knockout neuron. So again, the soma size is clearly increased, and the dendritic arborization is much more complex. And this is nicely shown here with the Scholl analysis, showing this arborization and affecting both distal and apical dendrites. When we look closer at the dendrites, we can see that they're thicker, and even the spines are much bigger in the knockout condition compared to the controls. So Charlotte also performed some whole cell patch clamps, and to assess whether those neurons are more hyperexcitable or not. And actually here, the results were less clear, but she could see an increased amplitude of the spontaneous excitatory postsynaptic currents. So how this may explain epilepsy is still unclear. So now the next question was, can this 1% of CRISPRized neurons sufficient to trigger spontaneous epilepsy? And the answer is yes. So we have implanted several mice with intracranial electrodes. And after 2 months of age, mice develop seizures, but in a very particular pattern. They have a cluster of successive brief seizures, and they end with a SUDEP-like event. So there's a suppression burst EEG, flat EEG, and the animal dies suddenly. So not all mice have seizures. Only 30% of those mice presented these seizures, but I guess this is mostly due to the variability of in utero electroporation experiments. So Theo then assessed whether rapamycin, so the mTOR inhibitor, could prevent those seizures. We have shown that when rapamycin is administered prenatally, we can prevent some migration disorders, but can we prevent seizures as well? So here he administered rapamycin at E15 for several days, consecutive days, and then he checked whether those mice by video EEG had seizures or not. And apparently this had prevented epileptic seizures for at least some days, although we suspect that there might be a recurrence of seizures once rapamycin is stopped. When we use rapamycin at adult stage, so migration disorder has occurred and large cells have occurred, we administered rapamycin. Again, this has prevented epileptic seizures. So clearly seizures are directly linked to mTOR activation here. And the role of the neurodevelopmental aspect is still a bit enigmatic at this stage, I have to say. In the lab, and this is the work of Alexandre Bach, postdoc, we have also generated some neuronal SYN1 conditional knockout mice. And as expected, because it's a stable line, I mean, they have a more robust phenotype. So 100% of the mice have spontaneous seizures around the age of 3 months, and all die of a SUDEP-like event. And you can see here the seizure and then the EEG being suddenly silent here. So we thought that we could use this model to investigate further the mechanisms of SUDEP. And I have to say that physiological mechanisms of SUDEP remains unknown, but there are 2 main scenarios. One being a cardiac arrhythmia, and the second one being due to a brainstem depolarization leading to a cardiorespiratory arrest. So here we aim to assess whether DEPDCS5 conditional knockout have cardiac abnormalities. We did that first because DEPDCS5 is well expressed in the heart. This is something that Alexandre has shown. And also, and that's the important point, because cardiac arrhythmia can be possibly prevented for the patients. Again, DEPDCS5 mutations have a higher risk of SUDEP, and with this, the idea was how can we prevent the SUDEP? So Alexandre has first tested this hypothesis using anesthetized animals, and he could show at the basal level that the heart rate, the QT and QRS interval of cardiac action potentials is normal, as expected. So he then performed combined EEG and ECG monitoring in live mice during the seizures. And actually, just before the seizure and even after the seizure, the ECG was quite normal, excluding a cardiac abnormality in those animals. And in parallel with Fabienne Picard in Genève, we also investigated 6 patients with DEPDCS5 mutations, and among the 6 patients, 2 have since died of a SUDEP. And so we did a cardiac investigation of these patients with Holter, echo, and ECG. And we also excluded a major cardiac alteration in the DEPDCS5 patients. So most probably, and this is what we are now pursuing, it's a brainstem spreading depolarization that is the cause of SUDEP. So now let's again have a bit a summary. There are many FCD mice models that have been generated. Each team working on a specific gene. So we have AKT3, mTOR, TSC, red models, the DC5, TSC1, TSC2, etc. And these, all of these models that are either targeting positive regulators of the pathway or negative regulators of the pathway will lead to migration defects to enlarge phospho-6 neurons. And most of these models, and all models, have spontaneous seizures. The particularity of the SUDEP seems to be specific to DEPDCS5 model and DEPDCS5 patients, unfortunately. And the only thing that these models did not recapitulate is the presence of the balloon cells. So mice do not present, apparently, balloon cells. So now just few conclusions. So we and others have contributed to elucidate the hidden genetics of FCD by showing a major role of somatic mutations arising during neurodevelopment and only in a subset of cells. And it is now very clear that all FCD type 2 are mosaic mTORopathies linked to an abnormal activation, constitutive activation of the pathway. We have also been able to demonstrate the occurrence of an SN2 hit mechanisms in FCD, and again, this has been now confirmed by other groups. And we have shown that the mutation load is enriched in dysmorphic neurons and balloon cells, so the mutation has occurred in their progenitors, and this is the cause of this cellular phenotype. We are also now armed with a very good preclinical FCD mouse models, which recapitulate nicely both the neuropathological aspect and the clinical aspect with seizures and SUDEP. So now what are the next challenge and the future directions, to my point of view? Well, the first thing is that, okay, it's great. We have elucidated the genetic cause of FCD, and it's great in terms of basic science and understanding about the neurodevelopment and the link with mTOR. But how can this impact the clinical care of the patients? Because so far we have a genetic diagnosis only after the surgery. And the question is whether we could do that before the surgery, and whether knowing what is the mutation and the gene mutated before the surgery could guide us toward the surgery outcome. Because one thing that I didn't say is that all patients are not cured after the surgery, and in 30% of them, there's a seizure recurrence. And of course, these genes are also going to guide toward targeted therapies, and I will come back to this in the next slide. So the idea is, can we detect those mutations before the surgery in biofluids? And this could be done in CSF and plasma. And the idea is that they would be released from cell-free DNA circulating in those fluids. What we are also aiming to do is to perform patch-seq in those human FCD to have a correlation between the morphology, the transcriptomic, and the genetics, and assess the developmental origin of these normal cells. But high-throughput single-cell profiling of these FCD tissues would also be very useful to identify biomarkers, specific biomarkers of dysmorphic neurons and balloon cells. And by identifying their molecular identity, we hope that we can then design therapeutics targeting specifically those abnormal cells. So we are not talking about hyper-precision medicine. And for this, we need to do some single-cell profiling and identify which clusters might correspond to balloon cells or dysmorphic neurons. And if some of you are interested with this work, we have 2 postdoc positions available in the lab. Now, this is, I'm going to just to end with few words about those targeted therapies. And so here's the challenge. We have in FCD field an unmet clinical need because FCD, but also GATA1 related epilepsies are drug resistant. to anti-epileptics that are commonly targeting ion channels. And so there are no etiology-specific anti-epileptic drugs, and this is probably the reason why ion channel anti-epileptic drugs do not work in those patients. So surgery can cure a certain amount of patients. However, it's of course very invasive, and some patients are not eligible to surgery because of surgical inaccessibility and also surgical failures. So there's still an important need to find targeted therapies. And with FCDs having mTOR gain of function variants, and this is the case for PIX3CA, AKT3, and mTOR, of course, there's the idea to give inhibitors of those involved in these pathways. So rapamycin, the inhibitor of mTOR, has long been thought to be useful and actually has already been used in several clinical trials, and in particular in patients with tuberous sclerosis. As I said, those patients have seizures, which are also drug resistant. And there was this very nice study published in Lancet a few years ago showing that rapamycin can indeed decrease the frequency of seizures in those patients, although it was not a huge effect. And rapamycin is not really an option to give to children in a systematic way to treat just a very focal neurological disorder. So we had also the idea, because the GATA1 branch is regulated by amino acids, whether a diet therapy could at least help and help these patients a bit in the spirit of the ketogenic diet, which is very useful in some childhood onset epilepsies. But my last slide is about this PIX3CA inhibitor. This is the work of a colleague, Guillaume Cano, working at Necker Hospital and Imaging Institute. So the team of Guillaume has been working on PIX3CA overgrowth syndrome. It's also called the CLOVES syndrome. And this syndrome is due to somatic gain-of-function mutations in PIX3CA. And those mutations are exactly the same that we find in a malformation of the cortical development, except that here they will occur in different organs and lead to different overgrowth organs. It's a bit the elephant man syndrome. So the team of Guillaume has developed a PIX3CA mouse model. And as you may see here, they have different overgrowths all over the body. And he then gave to this mouse a PIX3CA inhibitor here, and he could see that the overgrowth really shrunk in the mice. He has then been allowed to treat a cohort of patients, and this is one of the overgrowth patients before the treatment and after 18 months of treatment. The overgrowth has really shrunk. It was quite spectacular data. He has been treating adults and children, and since this publication, he has treating hundreds of patients with this. And so I think it's really a big message of hope about those therapeutic strategies. I mean, precision medicine is not only a term that we use to obtain grants, but I think it's really true. I am done with this, and I'm now just going to analyze people I'm working with, and mostly my team members, because they are wonderful and I like them a lot. Also the past members and people from the Fondation Rothschild for the brain tissues, and Larry Boisier for the neuropathology, and also many collaborators that I listed in a random order, and I'm sure I missed many of them, and I'm sorry about that. Thank you, and I'm happy now to take any questions.
Thanks, Steph. You can keep, it's up to you whether to keep the slides in case you need them or to sort of stop the share. There are a bunch of questions. I promised Steph that she can be out of here by about 6:20 because she has a train to catch. She has a brain also, but not to catch. Anyway, usually you shouldn't sort of correct your Freudian mistakes because you just make them worse. So I'm just gonna, there's a bunch of questions, Stephanie. I'm gonna read them to you in the order they appear in front of me, at least. The first question is from Amanda Almacelas. Sorry. Do you expect to be able to get balloon cells modeling the disease with in organoids? If you did organoids, would you see balloon cells?
Okay, well, so far, Is there an echo? No?
I don't know.
Okay.
I don't hear an echo.
All right. I can hear it. So actually, we haven't seen them so far. But we are using brain organics with DEPDCS5 iPSCs. And one thing that I didn't mention in the talk is that DEPDCS5 usually doesn't give rise to balloon cells. So I'm afraid we won't see those balloon cells in brain organics. Thanks for the question.
All right, next. So maybe the echo is coming from me having my mic on, so I'll turn it off when you are answering. Okay, the second question is from Maeva. Really interesting talk. Do you have any ideas of what would make the DEPDCS5 gene a hotspot for somatic mutations? My first thought goes to the transposon, for instance. Do you see any signature of transposition in that locus?
Yeah, excellent point. So far, we have no answer, no clue whether indeed there's a mutation signature, because in contrast to mTOR, PIK3CA, where we have recurrent variants, so we know that there are hotspots, in DEPDCS5, there are no hotspots. All mutations are different. Now, why is this gene more prone to be mutated? Or when there's already an existing germline mutation, why is it prone to get a second one? That we don't know. But that's a very good point to check for transposons. Thanks for the idea.
Okay. Next question is from Pablo M. Great presentation. 2 questions. Are these DEPDCS5 animals CRISPR or conditional? Do they show any other behavioral problems such as learning anxiety or sociability defects?
Yes, I didn't present this data, but this is the work of Alexandre Bach, and he has indeed investigated different aspects, anxiety, intellectual defects, aggressivity, and he indeed see some cognitive and behavioral comorbidities in these animals. And what is interesting is that he finds them before the seizure onset. So we can show that it's really an effect of the DEPDCS5 and not a consequence of seizures.
There was a second part to that question, which is if you treat neurons, so the DEPDCS5 mutant neurons, with BDNF, do you see an exacerbation of the physiological and or morphological problems?
No idea. We didn't do that yet. I'm sorry.
Okay, so
the idea is probably to increase the activation, right, of the pathway this way, but no, we haven't thought about doing that, but maybe we should. It's true that it's something that we discuss in the team, how if it's only the overexpression, the overactivation of the pathway that would lead to the phenotype, then we would be able indeed to address it by adding some other growth factors. So that's
yeah, so I had a related question to that, so I'll ask it now since it's so, you know, obviously the defect caused a sort of a developmental problem in terms of the migration and so on, but it also causes this morphological problem in the shape of the cells. And you were able to add rapamycin to rescue the developmental defect, but you didn't show us any sort of adult rescue. So in other words, if you turn off the pathway in the adult, even though the neurons are mismigrated, would they still be able to sort of readapt and function more properly, or is it too late? Is it really a developmental problem?
No, it's not too late. It's not too late. You can decrease in the adults the activation, and they can even shrink a bit, you know. So it's not too late.
Okay.
So there's really one off. This echo is very weird, huh? I don't know how it is for you, but I hear me twice. It's okay.
You were hearing it even when my mic is off?
Yeah, it's a bit weird, but it's okay.
Sorry about that. I don't know where it's. Usually, there's an echo if you're speaking and my mic is on, so people hear it twice. But I don't know on your end. Anyway. Yeah, so where are we? Here. Do you know if DEPDC5 targets mTOR, C1 or C2 or both?
I think it's targeting only the mTOR C1. I mean, at least according to the work of David Sabatini. It's, yeah. But I'm not sure this has been really addressed specifically with the mTOR C2 complex.
And then Alice Davy is asking, are the dysmorphic neurons and or balloon cells polyploid? Is it known whether they cycle or endocycle?
No, this is not known either. No idea. I mean, balloon cells have expressed some cycle proteins, mitotic cycle proteins. So we know that there might be a defect at this stage, but it's only immunostainings markers, right? There's no mechanistic work that has been done to really answer this question. We don't know, sorry.
Okay. Okay, so another question from Pablo M. Fasting reduces mTOR activity. Could it be used as a possible therapy too to reduce those seizures if people just fast?
Right. Well, actually, in some drug-resistant childhood epilepsy, ketogenic diet is used and can lead to clear improvement for these children. So diet therapy is not is clearly an option, especially for those who would not be candidate to surgery. So this is one project that we have in the lab, at least with the mice, to try to give them a specific diet. And especially because we know that leucine and arginine are the main amino acids regulating the pathway. We have had this plan for already several years to give them a specific diet and try this. So I think it's worth trying, clearly.
And finally, another question from Ayush Srivastava. So, if we try to target DEPDC5 in wild-type mouse pups, how long does it take to overexpress mTOR signaling in these targeted cells? Is it instantaneous? So, what is the time delay between inducing the mutation and actually seeing an increase in the activity of the mTOR pathway? If one could do that, for example, in a live experiment where you sort of induce, for example, a knockout with a Cree mouse or something like that, and then assess mTOR levels or P6, for example, at different time points. How long before you get to the activation? I think that's the question, right?
Right. Yeah, no, my guess is that it's going to be almost instantly, because that's what we see in vitro. I mean, we can increase or decrease the activation of the pathway in a few minutes. So my guess is that that would be very, very, very fast. Yes.
Okay. Well, we keep our promise. You can leave to catch your train before 6:20. Thank you, Stephanie, for a fantastic talk. Thank you, everyone. Stay safe, stay healthy, and see you here next time, next week, I guess.
Okay. Merci
Bye, everyone.
Ciao.
Bye.